Hello everyone. In the previous video we are talking about the application of the biotechnology and artificial selection. Now we want to move to other one application of the genetic engineering, of the biotechnology, which is genetic engineering. What is the genetic engineering? What is the meaning of genetic engineering? Here it's not like artificial selection. Here what it's gonna happen, the change in a gene or changing in the DNA to produce the desired characteristic. Hello, what is the biotechnology? New technology has made it possible to directly change the DNA of an organism. Okay, so the definition of genetic engineering is the process in which a piece of DNA, a piece of DNA, and you're talking about gene, with gene and is responsible to make a special protein by transcription translation. It will make a specific protein. So the definition of genetic engineering is the process in which a piece of DNA is modified for use in research, medicine, agriculture, or industry. Yani, if you want to talk about the DNA, for example, I'm going to show you this one here. This is a DNA, and as you know, each segment of the DNA is responsible to do a specific a protein, which is a gene. Okay, here if you change this D and it cut this, for example, this gene, and you replace this gene from other organism, here you're making what? You're making a genetic engineering. You're modifying the DNA. So scientists actually isolate, and where do they use these things? They are using this in research, medicine, agriculture, or industry. Scientists isolate and change a segment of DNA in an organism, or take a segment of DNA from one organism and transfer it to another. So this changing of the DNA, transferring from one organism to another, it's called what? Genetic engineering. Okay, let's look at this one here to understand the genetic engineering.
Genetic engineering is the process in which a piece of DNA is modified for use in research, medicine, agriculture, or industry. Click each image to learn more about this type of biotechnology. Scientists can isolate a segment of DNA in an organism, change it in some way, and then place it back into the organism. This process is also used to insert genes from one organism into the DNA of another organism. The new genes provide the organism with a desired trait, such as resistance to pests, or larger size, or fewer seeds. Genetic engineering can be used to insert genes into crops that give the plants desirable traits, such as being hardier, more resistant to pests, larger in size, or brighter in color. The tomato on the left has been genetically modified to resist mold, while the tomato on the right has not. Gene therapy is a technique that is used to correct a defective gene in an individual affected with a genetic disease. This technique is still in its experimental stages. People who are affected by type 1 diabetes do not produce their own insulin and must receive insulin shots. A form of E. coli bacteria has been genetically modified to produce insulin. To do this, the gene that makes human insulin is inserted into the genes of a strain of E. coli bacteria. After the gene is inserted, the bacteria becomes an insulin-producing factory. Once produced, the insulin is extracted from the bacteria, purified, and ready for use by humans.
Okay, I'm going to show you another video that help you to understand the genetic engineering. Look at this one. Listen carefully.
What is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result. To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes, code for certain proteins, which in turn make up all the different parts of the organism. Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent. All living things use the same genetic code, DNA, made from the same 4 bases, so scientists can use special enzymes to cut parts of the DNA from one living thing and sort of sew them on to the DNA of the target. The result is then a living thing that has a new gene and a new feature as a result of that gene. Now, there are many examples where genetic engineering is used today, but we're going to focus on a very specific medical example to understand this concept better. Humulin, a medication used for diabetics, is created with genetically modified bacteria. Humulin can be given to diabetic patients who lack the insulin they need. Now, how they do this is scientists figured out which section of our DNA codes for insulin, and they also found certain enzymes that can cut that gene out. What they do is, using enzymes and heat, they isolate a specific gene and then insert it into the DNA of bacteria. The result is now a bacteria that produces human insulin. And since bacteria can multiply really fast, you can create a lot of insulin in a very short amount of time. Now, this is great! Genetic engineering poses huge potential in the field of science, providing us with new insight on how our genes work. However, there's also a lot of controversy in certain respects to genetic modification, specifically genetically modifying our food. To learn more about GMO foods, check out our next video.
Okay, so this I hope this video help you to understand how to make the genetic engineering. For sure, it's not a 1 step or 2 steps. It takes a long time to do it, and it's not easy. For sure, you have to do experiments to make sure how to do it. Okay, but this is the main idea of the genetic engineering. So this one here, I'm going to solve. You have it here on your book, which is on page 164. You have a question, for what purpose can genetic engineering be used? Actually, we can use it, we said, like 4 things we can use the genetic engineering for. You can say, for example, for industry, for medicine, let's say agriculture, and let's say for we wrote it industry here, oh yeah, and research. Okay, so these are the for using. You have also in your book, I want you to look here. Look at this figure. And this figure here, it shows the plant cell, plastid, and this one here, actually here scientists have disabled a gene in the mouse on the right. As a result of this, mouse cannot control how much food it eats. So this one an example. This is another example here. These genetically modified plant cells produce tiny biodegradable plastic pellets. That pellets are then collected to make a plastic product. Okay, so these are example. This is an example of using of genetic engineering. Here you have a question. Traditional plastic are made from petroleum, as you know, a non-renewable resource. What benefit could plastic made by plants have over traditional plastic? Actually, plastic grown in plant cells would be more Sustainable because plants are a renewable resource, right? So this is how to use an example how the genetic engineering is good. Sorry for this. Let's write here the answer. Here the answer: plastic grown in plant cells would be more sustainable because plants are what? Are renewable resource. Okay, so this is all about the genetic engineering. If you have any question about genetic engineering, you can send me your question. Thank you for listening.